the utilize of the microorganism in the industry. So you remember the first part, we actually talk about the microorganism and how it's involved in the food industry, that various food that has been manufactured utilizing the microorganism. And today we continue. We talk a little bit more about that, the industry of microbiology, plus a bit of the others. Okay. The content. So, not very long, 3 parts. The first part is the major product of industry microbiology. As I mentioned, we will talk a little bit more and how that the microbes that contribute to the manufacturing of different type of product other than the food product. And then we come to the biofuel, right? And then the biodegradations and bioremediation. This 2 part is involved in both the environmental.
Hello, Dr. Huang. There's no audio.
That's why I tell you, never why you want to do online. I never encourage. And you're telling me that no, we have to have online this and that. Why don't we have just have a physical? That's too much. I'm going to collapse. Alright. I wish everyone can be physically appear. Okay. Oh, I'm not sure that I can turn on that. Now you have the audio, right? Oh yeah, we can continue. So, this too is about the environmental, that how the microorganisms relate to our environmental issues. And the first part is more on the manufacture of the product using the microorganism. Alright, the next one is your learning outcome. It's also based on the content. It's very simple. That you need to know what are the various industry products that utilize the microorganism, and at least you need to describe a few of them, not in the very detail, not in like how you're going to make the process, the detail, but you just need to know to name it and what kind of microorganism has been used. And then you need to know briefly how is the productions of biofuel. There are 2 biofuel that we will actually bring it here in this lecture. One is the ethanol, the other one is the biodiesel. So it's true that you need to actually briefly describe the process of making the biofuel. Last one is to describe the step of the sewage treatment. So sewage is the wastewater. So it's very important that that microbe has been used for the treatment of the wastewater. Alright. Now, in the industry of microbiology, when you try to manufacture a different type of the product, those are the 3 main categories that utilize the microbe. We have about the food industry, we already talked about a lot, but today we're going to include a few more, which they are those that are highlighted in blue. And also for some of the medical or pharmaceutical product, the one that I will highlight will be the antibiotic. And the biofuel, that's only 2 will be mentioned. One is the ethanol, the other one is the biodiesel. Alright, so I will only talk about those are highlighted in blue. Okay, the first one, how do we make the organic acid? We know that organic acid, especially like citric acid we can get from fruit, lemon, lime, oranges. The other one is what? The grapefruit, yeah? They have citric acid. So, this citric acid, which actually is very being widely used in the industry, very useful. We will see a few of them later on in the next slide. And to make it, basically you can just take in the fruit and extract the citric acid out of the fruit. But why we need to actually use the involve using the micro to produce the citric acid? Basically, it is because using the microorganism is economic. They are easy to grow, and they are cheap, and they make a large amount of the citric acid as compared you using ton of the fruit, like the oranges, but you only making a very small amount of the citric acid. So ideally now, no one actually are extracting the citric acid from the fruit, but instead of using the microorganism to produce the citric acid. It is a weak organic acid, and normally those microorganisms undergoing fermentations at pH 3 to 6. So it is a low pH, not high pH, and these are the microorganisms that normally used to make their citric acid, especially the aspergillus. So you have yeast, fungi, as well as bacterial. Arthrobacter and Bacillus are two bacterials has been used to make the citric acid. So how is mixed with citric acid? This is a very simple process. You have glucose. Glucose can be coming from polysaccharide, sugar, or starch. Right? So it is converted actually by the aspergillus. Let's say this is the yeast that actually can fungi can make it change it into pyruvate, and then pyruvate will make the oxalo, acetate, and acetyl-CoA. With this product and then combine, then they will have making the citric acid as the end product here. So the whole process will be the fermentations process. Alright. Now this is how that citric acid involved in the different kind of the product. Cheese, they contain citric acid. It's also been used some kind of the preservative, and the bread contains citric acid. Here the drink also contains a small percentage, and then this citric acid just makes the taste better, giving you the tartness of the fruity taste. Right? You also have here, what is this? This is potato chips. Also have the citric acid containing. In terms of the pharmaceutical product, and you have this, what is that? This is the cosmetic, it's a scrub, face scrub that is actually using the citric acid. I think you can see clearly here. Where that citric acid can effectively remove the dead cells. That's why it's included in the face scrub. And here, this is the test tube. Normally, this test tube we use to collect the blood, from patient. And you have different type of the color of the cap, indicating what is the purpose of the tube. And this tube is contains sodium citrate, 3.2%. Right? So this citric acid or citrix or sodium citrate is to prevent the coagulation of the red blood cells. So as always include in different. And here is a rust remover because it's acidic, and it can actually remove the metal oxide. Whatever is become rusty, you can use it to remove. So actually it contains in the rust remover. And this is a fertilizer. This fertilizer, the citric acid included because it's going to lower the pH as part of the component of fertilizer. So citric acid has been widely used in a different field as stated here. Now, we have the organic acid, as we we talk a lot about citric acid. Acidic, lactic acid is coming from, you found it in where? Yogurt, cheese, and then some pickles. They contain the lactic acid. Fumaric acid, normally you found in some kind of the food additive, right? How do they make the citric acid as an example? And very simple diagram, not too complicated. You have the Aerogenous niger, which is one kind of the fungus, and you having this bioreactor with its submerged fermentations. It's called submerged because the microorganisms actually are inside the medium. So it's not on the surface fermentations, it's a submerged fermentations because it's covered with the medium. And you're adding the nutrient, which is sucrose here. Where do you get the sucrose? You can get the sucrose from different as a substrate that is coming from beet molasses, black extract and molasses, or all kind of molasses, meaning that extract from sugarcane, cornstarch, and hydrolyzed starch, all this wood, heavy cellulose, olive oil, palm oil, soya bean oil. So all this actually eventually can convert into glucose and then into acid, citric acid, undergoing the fermentations inside here. It depends on what type of the microorganism that you use. Alright, another species that in use is the yeast candida that also produce quite certain amount of citric acid. And normally you're using the Aerogenous niger, which actually can grow in 2.5 to 3.5 pH. Alright. The second one, which is also organic acid, is called here hyaluronic acid. Very important, especially in the field making the cosmetic product. Hyaluronic acid present as a low molecular weight, where it is in the monosaccharide or disaccharide. So it's very simple. However, when you're actually making a hyaluronic acid polymer, and you become a very long chain, and it is having a very, very big molecular weight. And it will become like a jelly or gel-like structure, very sticky and viscous. So these are very useful. One way is that we will talk about that in the next slide. And here, just to show you that we also have the hyaluronic acid in our body. And mainly we found them in eye, the jaw, and also in our skin. So let's say that this is our skin. You have this is a cell membrane, this is an extracellular matrix. So you found the hyaluronic acid, basically is secreted. For example, this is a fibroblast, the skin cells, that they will secrete the hyaluronic acid into the extracellular matrix. So this is part of that, they conjugate with the proteoglycan, some kind of a sugar. Alright, they form a mesh. And why it present in our skin? Because the hyaluronic acid can retain the water. So therefore they say that 50% of the total body, right, of the HA, this hyaluronic acid, are found in the skin. So it's keeping the skin's moisture and retaining water. And when you're getting old, people found that the content of the hyaluronic acid in the skin reduced the level, the level of that reduced in the older people as compared to the youth. And since it can retain the water, it makes our skin's moisture, and it has been very useful in use in the cosmetic product. And you found here, let's say that here, actually do you found, yeah, you say that hyaluronic acid serum, And also this is the face serums that it contain at least 2% of the hyaluronic acid inside the serums. So it has been used in the cosmetic product because it's very good and useful to retain water to keep our skin moisture. However, when you're actually making using the hyaluronic acid for making the cosmetic product or using it as a lubricant for the joint, they usually using a different kind of the microorganism and making the high molecular weight and low molecular weight. For example, here, if you use the Streptococcus abitibuscus, actually they can produce the high molecular weight of the HA, and this is normally used for the lubricants in the joint. So you know the joint, you have osteo. Most people when you're getting older, they will have the, especially women, will have the osteoarthritis, meaning that you have the joint that you have this collagens and actually getting thinner, and you start to actually have the collide between the bone. It's very painful. So you have the knee pain. Alright, and also your kneecap, because there is a cavity here. So the cavity normally can keep the hyaluronic acid not easily to, because high molecular weight not easy to picking out. So it can keep it for, you may tell, the doctor may tell you it can keep it for 2 years, the most maximum, and then you need to go back and have another injection. Alright. If you have this Bacillus subtilis, they produce a lower molecular weight of the HA. So it is normally used for the cosmetic product. And even you see this one, right? This is the Streptococcus. It looks like the colony on the agar plate. It looks very watery because it can absorb the water. So it looks a little bit watery. So other microorganisms can also be used, such as E. coli, Agrobacterium, Lactococcus, or yeast, like Pichia. They all can use to as the alternate for the producing the hyaluronic acid. Alright, this one is amino acid. As we know that amino acid, we also use it for part of like a food supplement sometimes. For example, in potato chips, or is potato chips for the taste. What is this? MSG, I think is written. Right, this is the MSG. And also, sometimes of the, what is it? This one, the phenylalanine, aspartic acid, you found in the sweetener. And lysine or methionine, you find in the cereal food supplements. And MSG, I think that nobody would not know what is MSG, right? Everyone know. And the MSG, those company has been telling you again and again, MSG actually is coming from the natural product. It's not something synthetic chemicals, so it's very safe to consume. In actual fact, it's safe to consume. It wouldn't cause cancer, like what that has been. People have been talking about that, like 30, 40 years ago when it first come out. A lot of people using the MSG for cooking, and then they start to have it causing cancer. But in actual fact, it is very coming from the natural product. And we're going to talk about how we're going to make the MSG. How do you go in is easy. You extractions from the plant, such as a seaweed, and then you can, the way of doing it, you can do the extractions to get the amino acid. You can have the chemical synthesis for amino acid, and finally here you can actually also make it in the large scale productions for using the microbiological synthesis. Now, this is the one. Where should I go to? So this monosodium glutamate is actually a sodium salt of glutamic acid. Glutamic acid is one part of amino acid. You normally found this monosodium glutamate in tomato or cheese, right? And now, how did actually this has come from? This diagram actually originated from the Ajinomoto, the website. They are not telling you in the detail, but they will telling you the main process for the MSG. So they start to get the sugar cane because why they getting sugar cane as a feedstock? Because sugar cane has a lot of the sugar, so it can provide the glucose to the microbe. So then you're actually extracting the molasses and then you add it into the bioreactor. In addition, you're also getting those microbes to adding into the fermenter or bioreactor. They did not actually review what is this, the name of, actually they haven't, but exactly they did not review what is exactly they put inside the reactor. But there are two species here which actually can be used to making the MSG. So inside this fermenter, what happens is that you have the sugar, all these microbes actually absorb into the sugar and they has utilized and they are undergoing growth and then they release the glutamic acid. And then the glutamic actually collected in another big tank and then they further process it and crystallize it to dry, crystallize for 2 days, 20 degrees Celsius, and then further drying using the heat and then they pack. And this is the product of MSG. Alright? Vitamin. Vitamin, we know that vitamin is something supplement for us. So you can buy vitamin in the chemist. A lot of people taking it. I'm sure that you're also taking it. B2, B12, vitamin C, that is a water-soluble vitamin. Those are non-water soluble, A, D, K, A, D, E, K, I think. Is not the non-water soluble. And those actually, of course, you find vitamin in the food, but if you need additions, of course, you can make them, you can buy. And since there is a demand, so they actually manufacture the vitamin. So those are the table, and these are the microbes that can be used to make those vitamins. All right. Just give you a brief idea. What is it? Enzyme. Now, enzyme also can be produced. Enzyme used in a different way. Let's make an example of this. This is a detergent. It's just to remove the stain. So in the label at the back, you will have to find the enzyme. This is because they actually emphasize this is something plant-based. It's from natural. Okay, the ingredients are from the natural product. So you have this protease, amylase, lipase, cellulase, pectinase, and mannanase. Those are degrading the protein, degrading the lipid, degrading the cellulose fiber. So it is very effective. Enzyme is to use to remove the stain. Now the protease has been used in detergents mostly because we have the stain mainly coming from the protein, coming from food. That's why it's coming from protein. And you also use this in this bacillus called Bacillus amyloidoinfections. And amylase is another one. It's more to using to make the syrup, the fructose syrup. And here also this Abuginus niger is also making the glucoamylase. So in a way that they're producing this glucoamylase to actually break the starch into glucose and then convert the glucose into fructose and become the syrup. And the last one, enzyme, which is not food related, but this is a Taq polymerase. I'm sure you heard about the Taq polymerase. It's an enzyme that's normally we use in hospital and also in the research lab to use it for PCR. So polymerase chain reactions that utilize this polymerase because this polymerase is very special and can actually tolerate high heat. So even at 90 degrees Celsius, it won't actually degrade. So this polymerase has been very useful for PCR. PCR are useful for a lot of diagnostic method. So, there is a demand of taking this type of polymerase. However, it's originally from a thermophilic. I think this is a bacterial, not the archibacterials. It's called Thermus aquaticus, isolated from the Yellowstone in the United States. But of course, later on, a lot of archae or bacterials, they also contain this kind of polymerase. All right, very useful. Now, antibiotic, and everyone know antibiotic. Antibiotic are taken from penicillin are from the mold, right? So penicillium is taken from penicillium. And later on, they also found that the soil bacterial called actinomycin also making the antibiotic. So especially this strain called streptomycin is very famous that they're actually producing different type of antibiotic. So you can actually isolate a streptomycin. People have been looking for a new antibiotic, but somehow the first thing they would do is to actually explore any soil bacteria that contains streptomycin and try to find a new strain of streptomycin that able to make their new antibiotic. Of course, now we are not taking this approach anymore. All right, so we have this, because the current issues of using antibiotic, we are not allowed to simply buy it from chemists, first of all. If you go back to 6 years ago, you can get the antibiotic from any chemist, I think. Is it 6 or 10 years ago? I remember I purchased that before. Easily you can buy it, but now it's a prescriptive drug. You cannot simply just get it. And because we're overusing of the antibiotic. So a lot of bacteria start resistant to antibiotics such as MRSA, methicillin-resistant Staphylococcus aureus. So this is very famous because this MRSA has been shown to resist to a few antibiotics. And here, because the reason that MRSA, they actually has these transpeptidase. You remember when they're making the cell wall, the Staph aureus making their cell wall, they have these transpeptidase. You go back to chapter 2 and help to build the cell wall. But however, they start to have the mutations. So on this transpeptidase, therefore no longer, let's say the penicillin, beta-lactam family, the penicillin family, are no longer able to target on this because of this has been slightly mutated. So it's reduced affinity to this beta-lactam antibiotic. All right. So that's the problem. Now, currently, of course, not a lot of people are looking a new way of making the antibiotic. So you will have the progressions showing here, and 1, 2, 3 step. We are currently at this stage. So at the very beginning, the first generations of the antibiotic coming from the natural antibiotic. Simply that the antibiotic is making from streptomycin when they're undergoing fermentations. Let me see the next slide. Okay. Then you come to the second stage or second generation of antibiotic, which is a semi-synthetic antibiotic. In here, actually they combine the first generations, how do they make the antibiotic, and then they modify the antibiotic which is naturally produced. So they become a semi-synthetic antibiotic. And then in the third generations, what we are doing now is to synthesize totally a new antibiotic out of nothing, basically. Are not those antibiotics are not no longer derived from any microbes, any microbe that produce, we synthesize it ourselves. Okay, we talk about the first generations. So very simple. The first generations is penicillin is the one. You actually grow the penicillin in the fermentation tank, and then you make them undergoing. Remember that antibiotic is a secondary metabolite, meaning that they only produce when they are coming to the stationary phase, right, as a secondary metabolite. So they grow here, and then they collect the penicillin, and then they make product. Now, people has been looking for a new strain that produce new antibiotic. How people doing it is that if you want, at that, I think like 10 years ago, I know someone is doing it. They just go to any oil pump, the field, and then they collecting any soil, different type of soil, because inside the soil they may able to isolate the streptomycin. So they collect it, and then they just culture whatever, extracting anything from the soil, and then they culture it. When they culture it, you have a lot of colony grow, of course. So they isolate each of the colony as a single isolate. So you will ending up with 100 of the isolate coming from the soil. So these 100 isolate, they identify is the streptomycin. It could be same strain, it could be different strain, and then they will culture these 100 over 100 isolate of streptomycin, and then each of them, after they grow, they tested on their other bacterials. So you will have seen something, I forgot what is the name of that. You will have a disc that you are having, let's say a penicillin that dropping on the disc, and then the disc putting on the agar plate where you have to grow a lot of the bacterial. So along the disc, around that, surrounding it, it will become a clear zone, meaning that the penicillin actually killing the microorganism, the bacteria that go growing surrounding the disc. Alright, they are using this one to test all the isolate, the extracting some extract, bacterial the extract from those isolate, and tested on different bacteria and see which extract actually can kill better, kill different strain of bacteria. In this way, they can actually identify a new antibiotic. And this is also in the very first time, the first generations, how they actually look for the new antibiotic. However, everything can be exhausted. They no longer find any new strain that produce new antibiotic. Now they start to thinking, let's say that if I can get some antibiotic, because and then we can make some changes, modify it, maybe you can increase the efficacy. Right? Not decrease, but increase the efficacy of killing the bacteria. That's possible. So they start to modifying the naturally produced antibiotic. So what happens is that they will actually have, there's one strain called Penicillium glycogenum that actually can produce all these calling 6-APA. 6-APA is 6-amino penicillinic acid. So this 6-APA produced by this penicillium is having a very is actually having a very simple structure of a penicillin. It's a basic structure. So if this is a penicillin G, let's say, then this is a side chain that can be, they have a different side chain. Alright? So they maintain the rest is the same. It's just this side chain can be converted in any other side chain. So when they do modification, they simply alter this side chain and they make it into different type of antibiotic. So that's the reason they actually use this particular strain to produce this 6-APA. And this 6-APA can modify, adding different side chain and as a result, giving you some different type of semi-synthetic penicillin. So penicillin that produced by these second generations contain the methicillin and ampicillin. And this one is a rifamycin. This is a natural one, and this is a semi-synthetic one. Simply that this particular side chain has been modified. All right. So second generations using the naturally produced antibiotic and make modification on it. And of course, testing the efficacy. Does it work well? Like this methicillin and ampicillin, they work well, and then they put into the market. Now, then third generations, and because they're out of options, then they have to go for something new. So in this case, they're no longer looking into any bacterial strain or any fungus that are able to make the antibiotic. They are chemically synthesized based on the structure, and all of these one, right, naming here are coming from the fully synthetic antibiotic. What do they do is that they have using a software, and the software allowed them to predict the structure. So let's say that I want this antibiotic targeting on the cell wall or LPS. Let's say the LPS of bacterial. So LPS, I know that we already know by now, we already know well all different type of protein structure from the bacterial. So we can actually have the protein structure dump into the software, and then I want to have synthetic antibiotic that allow me to bind to this bacterial protein. So I also have the structure that they created that able to bind to the LPS. So with that, I can ask the software, or you can call molecular docking, is a general name, actually to see two protein components, how do they actually interact with each other. So to generate maybe 1,000, I will say up to 1,000 of these synthetic antibiotic that can specifically target to the bacterial structural protein. Now, you have this 1,000. They are synthetic. Then you actually synthesize them chemically. Alright? And what happens is that, of course, you have them in the collections like this 96-well plate, and you're going to use it to test on the bacterials. So you will have the agar plate, as what I mentioned just now. You will have the agar plate, and all these synthetic compound, right? You will up to 1,000. You just has to test it out on the bacterial, whether they can kill the bacteria or not. So you will actually have a disc, a circular disc, and that contain the synthetic compound. So if nothing grows around this disc, indicate whatever this synthetic compound can kill the bacteria. So you're having a clear zone. So testing this 1,000, normally it will cut down, if you're lucky, you can find roughly about 10, which is work very well. So it can be further test as a new antibiotic. So how do they further test? Of course, they need to test preclinical trial just to test how useful. Let's say that they infect the mice with bacteria, and then they trying this synthetic compound on the mice. See how effective that this synthetic compound can kill the bacteria. Of course, you also have a clinical trial eventually. And trying on this, I think there is one more step here, maybe. You also testing on human cell line just to make sure that antibiotic doesn't cause any harm to the human cells before you go for preclinical trial. All right? So this is how the progressions of making the antibiotic. Now, we go for the, do you need to have a break? Okay, you have to have a break. I give you 5 minutes. 5 minutes. I'm trying to settle this. I tried to make the screen, but I really that's the speaker screen, but change. Can you still see the slide? I now in Alright, okay, so because I was running it as a slideshow, so I just create a slideshow and you just if I move the slide, oh, it's not moving on the side. It's not moving. See changes. No, right? Now you can see I have achieved because I put on the slideshow that actually not to have. I do not know why. Unless I open up like this, all the people will be seeing this without looking at the slideshow. Yeah. Okay, thanks. You can open your own slide, right? Just to let you know that this week we do not have practical class. Did you know about that? So somebody may come early morning for the tutorial and then go home. Yeah? So no practical class this week, but we will have it next week. Next week, no tutorial, I think. And you should already have received my notice regarding to your assignment. So within 2 weeks, you should go in to e-learn and do your assignment. Very simple. Is it 1 minute?
But it still makes sense because like let's say
5 Alright. Let's continue. So this is another part. We talk about the renewable resources. So what is renewable resources? That is a definition. Simply speaking is that the resources that we use can be replenished and you never run out. So what kind of resources can be repeatedly using it and you would never run out? So that's what is called renewable resources. So it's a resource that can be used repeatedly and does not run out because it is naturally replaced, such as solar, wind, hydro, geothermal, and biomass energy. So that is the definitions. So we all know that the resources used for our electricity or all kind of energy, right? Consumptions are based on the fossil fuel. So fossil fuel include the petroleums, coal, natural gas, nuclear, electric power. But we always talk about that green environments, sustainable environments, right? And therefore, there is an urge that we develop this kind of renewable resources. So looking at this two comparison in Malaysia, this is recorded in 2020, and this is how we actually the consumptions of the energy in Malaysia. Petroleums and any other liquid, I think we utilize that is 7%. I think the majority are using this to generate energy. The coal that we still having rely on it a lot, 21%. Natural gas, I think believe that Malaysia producing natural gas. That's why it also the usage is also high. And only 6% that we try our effort to actually develop this kind of renewable resources as compared to the United States. Similarly, the petroleums that 35% here and 37, only 2% difference. And the natural gas is 36%, 34% here, only 2% difference. And of course, they have the nuclear power. They have 9%, we do not have this. And the coal they're using is 10%, but we rely a little bit more, 21%. And some more, they also having this renewable energy that they consume is 12%. We have only 6%, half of what they are using. All right. Of course, they have much more detail inside this renewable energy. What are they? So actually, it's written here. So these renewable resources include biomass, which we're going to talk about that a little bit more later. Of course, it also include the hydropower. So the hydropower is having this hydropower plant that you use the water that running into the waterfall and generate the energy. The geothermal power, you don't see that in Malaysia. I think that because they're using the heat coming from the underground and generate the energy. So wind energy, such as these, and then the solar energy, of course, solar energy, everyone know about that. Okay, so what about the biomass? It's a new field that research is still going on. People is trying to make it. So biomass as definition is organic. It's made from a material that comes from the living organism, such as plant or animals. So they're using plant, animal as a feedstock and utilizing microorganism to convert this feedstock into something that to generate energy. All right. And the biomass include the algae, and from the small tree, agriculture, whatever the leftover, the grain, the glasses residue, even the waste can be used as a feedstock, raw materials to use for generate energy. Of course, the wastewater or sewage sludge can also be used. So biomass energy is all called bioenergy. It's energy that you can generate from plant or animal derived materials, such as those other biomass. Now, the biomass here, when this is a raw material we begin with, using the microorganism, we can convert this biomass into the energy. The energy, if you go into the liquid form, we call biofuel. There are two types of biofuel. Biofuel include the ethanol or biodiesel. So anyone can recognize this picture. It's from a movie. Back to the Future, good. I thought it's only old generation know about that. So what is the Back to the Future, this story, the film about? Is there? Talking about somebody from future, no, I think they go to the future, right? And then back again. I think it's about I watched this one when I was a high school student, so you know how am I. This film is produced in 1985, which is how many years ago? 30, 40 years ago, roughly. This is about 40 years ago, right? Yeah. So here, actually, this is a professor, whoever, I forgot his name, I forgot his name. He using his time machine from future, actually he coming from future, and then he come back, and using his car actually is a time machine, allow him to actually travel back and forward. So when he come back here, the first thing that he used, actually he said, I need the fuel. He need the fuel for his car. So he actually grabbed something from the garbage bin, some banana peel, and actually this is a beer can, and they throw into this, right? This is the engine, the car engine, and allow the car engine to all the waste that from the garbage bin that he used to turn into the energy for the car. So this is the idea. When I look at that, at that time, I was thought this is very funny. It's very creative, but it's very funny. But I never know that one day we can really use the waste to actually turn it into the energy. And this is what it is about, the biofuel production. Basically, it's using something we do not want, food waste, or any leftover from the agriculture, that we use it as a biomass, and from biomass, then we make into either ethanol or biodiesel. All right? We have talked about the very, very first, how we start making the biofuel. Ethanol as an example. So at the very beginning, people will think that, okay, we are using, because we know that all these agricultures, the plant, first, second, third generations. So in the first generations, they use those, right? The feedstock actually coming from the agricultural biomass, can be a grain, corn, sugar cane. That, because all this, they are contain either starch or glucose. So they can be used as a nutrient for the microorganism. Therefore, they has been using it, this kind of feedstock, and a very popular at the time, they're using the Saccharomyces cerevisiae, which is the yeast, to make the ethanol out of this agricultural biomass. Of course, another type of yeast, the Pichia or Proberomycin, can also be used as a microbe to make the ethanol. And ethanol, why we're calling ethanol as a biofuel? We don't synthesize ethanol. Synthesize ethanol, it takes quite costly. So we're using the microorganism to turn the things, the plant, into ethanol. It can be used to mix with the gasoline. So like 10%, I don't think that 10% of ethanol, 90% of gasoline, or even 20% of ethanol, 80% of gasoline to mix it, and actually it can reduce whatever, you use it as a gasoline for the car to reduce the productions of carbon monoxide, carbon dioxide, and also the smoke causing emissions, meaning the smoke and the fog reducing is less polluted. All right, so that's the reason they like to use ethanol as a blending agent for the gasoline, because it's making less pollutions. What else? I have read a report that Malaysia, we don't make ethanol as a biofuel because it's not feasible. But we do make the biodiesel as a biofuel, but not the ethanol. Right? That's why you do not, in the petrol station, you don't see any ethanol that actually label that. You have seen the biodiesel, but not the ethanol. they make the ethanol out of the sugar cane or the corn. Now, is this a process, right? I think it comes from piece of yeast, eventually come for the fermentation time. So they're starting with the corn, they collect it. Of course, they need to grind the corn into small particle or even a powder. So they go for the mirror, and then they grind into breaking the top, coating into very smash and slowly. And what happens is that then they go for this so-called liquefactions or centrifications. So liquefaction and centrifugation is to, because it's coming from starch, let's say, the starch from the corn is a polysaccharide, right? Very big in molecular weight. And it normally cannot be consumed by any microorganism. So in this step, they actually breaking it down into small change, eventually to glucose, that it can be transported to the fermenter and used by the microorganism as a nutrient. So this is to bringing up the long chain to the short chain. It can be processed at high temperature. If it's high temperature, they're using the heat to break down the polysaccharide. If this is carried out at low temperature, they are actually using the enzyme to break the long chain starch. Okay, so one, it actually become a glucose that you actually input into the fermenter and also dump into the, let's say, the yeast, and then they undergo fermentations, and then sugar can convert it into the ethanol. Centrifugations is to try to remove all the debris, such as the debris from the bacteria, the yeast cells. So to centrifuge it, to remove and only collect the supernatant or the solvent out of this. Distillations is used to purify the ethanol. So there will be a few steps in here to purify the ethanol until it's more to 99% purity. So once you have that, then you can actually mix with it as a biofuel that can be mixed with the gasoline. Alright, so this is how it produced. Now, you come to the second generation. They are still using the similar process, similar method to produce the ethanol out of the feedstock. However, the feedstock they're using are no longer sugarcane or corn, because sugarcane or corn is actually can be a food for human. So that is a waste and also quite costly. If you're supposed to be a food, but you turn it to use it to make ethanol. And now the second generations, ideally the method here, they still remain the same, but the raw material, they are no longer using corn starch as a starch, or they're using something that, which is something that left over or something are not, that human not using it as food, but cassava is some kind of ubi. I think we are still eating, I think Asian, but not in United States, I think, not in any European country, but in Asian, we are actually having this as a food. So cassava is something very cheap that people not eating consider. And Miss Candace is something that is also we are not eating. A straw from the paddy field, glass or wood, that's something left over, right, from the agricultural farm. Those are the waste that we use it to make the ethanol. However, that's also drawback, because in order to actually process this, you have complex processes. So it's not like sugar cane, easily you get the sugar out of that. But this has a very minimum of sugar. So in order to actually extracting the sugar, the process is very complex. And you need a special skill, and you need more labor to do that, and high production costs, because I think the step of getting the sugar out of this actually is have to go through many steps. So it's a high cost and also short shelf life. Alright, so this is not easy. So then they abandon it, and they go for the third generations, which we are doing it now. So this is a third generations biofuel, and I'm using the biodiesel as an example. In the third generations, I think currently they have still very hot. People are still working on it, research on it, to actually find a good way to using the microalgae or very small other microbe to produce the biodiesel. So microalgae is having this group, consider microalgae, blue-green algae, which is cyanobacterials, green algae, dino, red algae, brown algae. Red and brown algae, they can be very big. Green algae can be small, like in the micrometer in size. And of course, the blue-green algae also very small, cyanobacterial. This is cyanobacterial. And this is a farm, microalgae farm. You see a lot of them. They are growing the microalgae. And this is a close-up of that. So they look very green because they are growing the microalgae. And how do they make the biofuel out of the microalgae? Something that now here. So as a taking example of cyanobacterial, and of course cyanobacterial require very, I think is very economic in the way that it doesn't need to feed any nutrient. They do photosynthesis, just need the sunlight. Right? We grow the microalgae, the cyanobacteria under the sunlight, and then when they grow, and they already find the strength, it's very strength, which type of strength of microalgae or cyanobacteria is very important, because microalgae, they can making the lipid. They have the vacuole, and there is a lot of lipid that accumulated inside the vacuole. So this is the cyanobacteria with the vacuole and the lipid. When they grow, certain strength that produce a lot of lipid. Alright? So this is the conditions that for the growth, which is very important to manipulate the growth. So all of this that when they have growing in large amount, all the lipid inside the cells will be extracted. A lipid then will be converted into biodiesel. Okay, you get the idea? Why we want to get cyanobacteria or microalgae? Because we want to extract the oil, the lipid, out of that. And we utilize the oil coming from the microalgae and turn it into biodiesel. So it's very simple, the step that you actually process, how to process, cultivate. Alright, you have the cultivate with essentials, whatever, the sunlight nutrient, very minimum, water, carbon dioxide. And you harvest, you collect the cells, and then you remove the medium, and you only got the biomass only. You centrifuge, whatever the way that you use to harvest, you can use the centrifugations to separate the medium from the cells, gravity sedimentations, just to let it to settle at the bottom, or cell aggregation, just to let it to make the clump. Whatever, what do they need is the cells. Then you extract the oil from the cells. So extracting the lipid out of the cells. So the method to extract the lipid, cell disruptions, first of all, you can use the ultrasonic wave, meaning that it's an ultrasound that you can break the cells. So, and you can use an enzymatic way to actually dissolving the cell membrane. And then you separate oil between the oil and water. What we want to is to get the oil. Now, once we get the lipid, normally they will produce something so-called triglyceride. So then this triglyceride have to make into, convert it into biodiesel through the process called esterification. Esterifications. Alright? So how do they do that? This process is basically you need one triglyceride, which is coming from the microalgae, and you need 3 mole of methanol. So 1 to 3 portions. And with the catalyst, which is using the either is alkaline or acidic. In this case, it's sodium oxide, and as a catalyst. And you are moving this to swap with that. So this will combine with here, make it into the glycerin or called glycerol, and then this part will be combined with the triglyceride and it making into 3 fatty ester, 3 mole of fatty ester, and this is called biodiesel. Alright, this is how they make it into that way. Cool? So this is the biofuel. So 2 examples in given, ethanol or biodiesel. Both of them are considered as a biofuel. The next one, biodegradations or bioremediations. And in this part, I only focus on bioremediations. Now, this is terminology slightly different. Biodegradations is a process. So it's a process to break down the organic matter by the microbe. But bioremediations is considered a technology or a method. So when you call it bioremediation, you're actually referring that the technology that allowed to degrade the organic matter by the microbe. So it's a method that applied. Now, example given here is the sewage or the wastewater. Now, we are generating tons of wastewater every day in every household. From bathroom, from a kitchen, the drainage that come. In Malaysia, I'm not too sure. Do we have the sewage treatment plant? Yes or no? I think we have, but not in the KL. I think our water is drained here and then all the way it goes to the ocean, to the river and then ocean. Right? Now, it becomes a very, very serious problem, especially in a third world country. A lot of poor, they do not have the privilege of using the clean water. So what do they take the water from? They take the water from the river, from the pond. So let's say if they has been contaminated, so that they actually they've been infected with different type of disease. So it's a big issue, and even you go to the WHO the website, they also talk about that as well. So making the environment clean is very important. Alright. Now, for the sewage treatment, there are 1, 2, 3 steps: primary, secondary, and tertiary. And what is that sewage treatment? So using a microorganism to degrade the organic matter, whatever is this kind of organic material, it could be the food waste, whatever is coming, the drainage coming out from a kitchen, yeah, those organic matter, and can be utilized by the microbe as a food. They degrade those kind of organic matter waste, and this is called a process of microbial bioremediation. All right. Now there are 3. The first is considered, which is a physical treatment. The secondary, which is considered, is the bioremediations. So this is the part that using the microbe to digest all the organic compound. So the tertiary here, which is considered a chemical treatment to further to clean the water, the wastewater, before pulling back the water back into the environment. So 3 steps here, and mostly we focus on this step. So how are we going to do with that? I have a diagram that's showing nicely. This one, right? You have primary. This diagram only show you primary and secondary. Now, you have the sewage, right? The wastewater coming here. So the wastewater coming here is contain everything. So you need, at the very first, you need to have the screen bar, or call a screen bar, to actually block a big object. What kind of big object? It coming from the pet bottle. It can be your food. It can be clothes. It can be umbrella, shoes, any big object. No, not be surprised. You can find all those kind of objects in your wastewater. So that is a screen bar to block all this going further. So when the small particles or the water, the wastewater coming in here. So this is a grid chamber. So the grid still need to be removed. So this is a chamber that actually using the gravity to settle the bigger particle, the grid, and then removing the grid. And then this wastewater further down go into this so-called primary clarifier. So primary clarifier, again, they try to settle whatever the heavier object as considered at the bottom, becoming a sludge. And those separating you, in this part is just the water, and on top of it, actually what is that? There are some very oily liquid. For example, the oil. What kind of oil? Lipid, fat, oil from meat, whatever. So this part is called scum. So sludge can be feces, it will actually settle at the bottom, it will go into here, right? And only the scum, whatever very, very oily stuff, will be brushed away by the scummer. So only this center part, which is the water, wastewater, is further go into here, and it's called clarified water, and they go into this secondary treatment. In the secondary treatment, you will have the aeration tank. This aeration tank given a lot of oxygen. The reason is that is a microorganism, the bacteria here living different type of bacteria, and they require oxygen to grow. That's why they provide the oxygen, and water go into here. A lot of those organic matter will be converted into something else, which is less polluted. Alright? Now, wastewater come here, supposed to be slightly clean. Now they come for the secondary clarifier. So again, they settle down. So whatever settle here, they may be, this is called activated sludge that contain a lot of activated microorganism because it's coming from here. So they are trying to utilize this activated microorganism again. So they are sending it back here because it's reusable. So they're sending it back here because when you're actually flowing, the microorganism will getting less, right? So that's why they're collecting it back and sending it back, and to grow more microorganism to do the process. And once it is this water, the clarified water, after the secondary treatment, it will go for the tertiary treatment. Here, you will be treated with chlorine. Because this coming here, they will still have some of the bacteria. It could be escape, and then it's just come over here. And other than the bacterial cells, they also contain a lot of nitrogen, phosphorus, and those are the chemicals which actually not being utilized from the wastewater, but not being fully utilized by the bacterials, also come here. So here, they actually treated the water to remove phosphorus, nitrogens, and also killing the bacterial cells. I'm using the chlorine, I think. The chlorine that actually can kill and purify the water further. So after this tertiary, then they will going back to the environment. So the water coming out should be clean. Although you cannot drink it, but it is clean enough to going back to the environment. Alright? And this is how it works. But let's say that in some of the more advanced Basically, the wastewater, actually, when they're coming out here, it's become a drinking water. They are trying to do that as well. But of course, there are, in addition of the assisting the system, they add another filtration, a very sophisticated filtration, can filter out even very, very tiny bacteria cells. So whatever the water coming out is clean enough as a drinking water. Alright, this is not very important. It's just telling you that what is the biological oxygen demand. So when you have CDs, actually there's in percentage, meaning that as we know that if you want to actually measure how dirty, how polluted as a wastewater, you can measure the BOD. Because if the wastewater is polluted, very, very much not clean, of course there will be a lot of waste inside, and the waste considered as a nutrient for microorganism. So microorganism is going to actually take up those nutrients, and then they actually can produce a lot of oxygen. Alright? So the amount that needed for the oxygen by the aerobic biological organism to break down the organic matter. So it's measuring. So you have these two actually systems. This is the one that I just mentioned, and this is the one is so-called trickling filter. So this is a little bit classic. So you have the sludge, and you also have the clarified water. So how do they purify? They are purified are not using the aeration tank, but instead they are using this something like that, which they have a filtration. So they have a different size of the stone or sand. So it's actually through the filtrations, and the purified water are dumping from the spray from the top. So the water, the wastewater are going into the vertical directions. So when you go through it, there is a lot of microorganism actually is on the surface of this kind of the stone, and the microorganism, as the water pass through it, then the microorganism can convert that into something that clean. So whenever coming out here, right, at the end, they're supposed to be clean once they go through here. But this system no longer, this is a very traditional way of the wastewater treatment, but now we more or less using this. The reason is that it can cut down the BOD from 35% to 25%. So if you have higher BOD, meaning that you still have remaining the microorganism in your wastewater. So if you have lower BOD, that means your water has less microorganism, which is considered cleaner. That's why this is a better method, which is aeration tank, or it's called activated sludge. Is a better method for cutting down the bacteria or the microorganism in the wastewater. Okay? Good? Do you want to end the lecture?
Yes.
Not yet. The last one is a video. Alright, I think you can play. I hope that you can play.
Cities and towns have sewerage systems in place to carry the wastewater from our homes, hotels, factories, and other establishments to a sewerage treatment plant where the wastewater is treated. It is then deemed safe to release into other water sources. The treatment of wastewater goes through different stages. Wastewater is treated to remove the physical, chemical, and biological contaminants present in it through various processes. The pre-treatment process involves the sewage being sent through grids or vertical bars that can remove large solid substances like metal cans, paper, and plastic materials. In primary treatment process, the sewage flows through the grit chamber very slowly, so that the sand, pebbles, and soil settle down at the bottom. The sewage flows into the settling tank or sedimentation tank, where the solid wastes like feces are allowed to settle down. Wastes such as soaps, oils, and grease rise to the top of the wastewater. The waste material that settles down at the bottom is called the sludge, and the floatable material is called the scum. Scum is then removed using a skimmer, and sludge is removed with a scraper every few days. The water that is left out is called the clarified water. In the secondary treatment process, biological or organic wastes are removed. It is a biological process. This is done by transferring the clarified water into an aeration tank, where air blowers bubble air, which helps the aerobic bacteria to grow and feed on the organic contaminants such as food waste, feces, and other organisms. The mixture then flows from the aeration tank into the clarifier, where activated sludge settles out by gravity. The activated sludge so produced, along with the sludge produced in the primary process, is transferred to the digester, where it is decomposed by anaerobic bacteria. Biogas is produced in this process, which can be used as fuel or can be used to produce electricity. The activated sludge is then left in the sand drying beds. Some quantity of water in the activated sludge evaporates, and the remaining quantity drains through the sand beds. This leaves behind the remaining dried sludge, which can be used for making fertilizers or compost. In the tertiary treatment process, the leftover wastewater is treated with chlorine to remove the phosphorus compounds, nitrogen compounds, and bacteria. It is a chemical process. Chlorine tablets are added to kill the germs. This process is called chlorination. Then this water is let off into the water bodies.
Alright, that's all for the video and that's all for today's lecture.